Well, we're in for a treat. The first speaker in this session will be Arup Chakraborty, who's a professor at MIT. And before I introduce him, I got to say a few things about him. Arup is an institute professor at MIT. There's only 12 faculty at MIT who have that distinction. He's a professor in chem engineering in the School of Engineering and a core faculty member in physics and chemistry. Now, you have no idea how rare it is for a faculty member to both be a professor in the engineering school and in the School of Science at MIT. And we're just so excited to have him. Arup moved to MIT in 2005. He was a professor at Berkeley, and his a very distinguished career at Berkeley before he arrived at MIT. And he's been an amazing colleague here. He's also the founding director of the Institute for Medical Engineering, IMEDS, which is a program between engineering, science, and medical students at MIT and HST. And he was the founding director of IMEDS and did a spectacular job launching it. He's recently been the steering committee that navigated the founding of the Reagan Institute, which is a building going up about 2 blocks north of here. The Reagan Institute is focused on immunology and combining science, engineering, and clinical immunology in a variety of fronts with faculty with joint appointments at MIT as well as appointments at Harvard and Harvard hospitals. So he's had an enormous impact. So to build the pressure on this lecture, I have to tell you that Professor Chakraborty won multiple teaching prizes at Berkeley when he was there. And he's won multiple teaching prizes at MIT as a faculty member. So, Arup, the floor is yours.
Thank you. Anyway, so I'm going to talk about an aspect of transcription, and let me further caveat what I'm going to say by saying that for 23 years, the major focus of my work has really been on understanding how the immune system works and then trying to harness that knowledge toward practical ends. But about 6 years ago, my colleagues Rick Young and Phil, whom you heard from, started to teach me about transcription. And so all the work that I have done in this area has been in collaboration with them. And so I'm as much a student as a teacher in this field. In any event, let me start by some history. And by my whole talk, I'll first give a lot of introduction to provide the basic backgrounds. And then I'll focus on the topic I really want to focus on. That is some specific aspect of non-equilibrium regulation pertinent to transcription. So by way of introduction, let me say that membraneless organisms have been described in cells for a very long period of time. And here, for example, from over a century ago, you will see references to organelles that lack membrane, have high refractive index, and seem to dissolve. E.B. Wilson in 1899 in Science noted that these things appeared to be like a mixture of liquids, and there were suspended drops of a different chemical nature. In more modern times, they have also been observed, but as I'll describe, without understanding really the generality. Here, for example, is a paper from 1999 in Science from Mike Dustin and others that concerned the discovery of the immune synapse. So what you're seeing in images here, and focus only on the bottom panel here, are images of molecules at the interface between a T cell, an important player in adaptive immunity, and a cell that presents antigen. And what you're seeing is two kinds of molecules, T cell receptors, shown in green, and adhesion molecules shown in red. And you can see as that time goes on, they form this body on the membrane that certainly is not surrounded by a membrane. And it is a well-organized structure with a much higher density of different kinds of molecules than before any stimulation happened. And then shortly thereafter, there was this paper that pointed out that, these essential characteristics formed by a spontaneous self-assembly process that were then active non-equilibrium processes were superimposed on it. And shortly thereafter, it also became clear that these were biochemical factories that played a particular role in concentrating all these molecules in one place, which allowed ubiquitin ligases to come and downregulate the TCR. But for all these observations, the major trouble is that no one understood that any of this is as general and ubiquitous and common as happened in a seminal paper and a pioneering paper, and a paper of great importance in 2009 by Cliff Branchman and Tony Hyman, who noticed and characterized that P granules are liquid-like droplets. P granules are made up of RNA and RNA-binding proteins. And then there was an explosion of interest in this field, which has led to the fact that we now think that many bodies in cells are biomolecular condensates. They may form by phase separation processes, and it doesn't have to be a liquid-like phase. It can be different kinds of phases. And these bodies can help organize molecules, concentrate them, for example, allowing them to conduct chemistry. In some sense, they can be biochemical factories, and they can dissolve when their purpose is done. What I'm going to do today is talk about these sorts of bodies, but in the context of transcription. Now, transcription lies at the heart of life itself. For example, during development, cells in a multicellular organism, different cell types emerge, which have different functions, and they are distinguished by having different transcriptional patterns. Some genes are silenced, some genes are active. Just to keep everyone on the same page, enhancers are non-coding regions of the genome that help recruit transcription factors and various other molecules in the transcriptional machinery, such as co-activators, RNA Pol II, etc., to near the coding gene, which then enables transcription of this coding gene. Now, a few years ago, Rick Young's lab noticed that the genes whose expression is critical for maintaining cell identity, that is, this is a heart cell, or skin cell, cancer cell, those genes are regulated by large clusters of enhancers, which are called superenhancers. And these superenhancers are characterized by a very high density of these transcriptional machinery. They are very sharply sensitive to perturbation. For example, they're much more sharply sensitive to drugs that can inhibit transcription. Taking away one element of the enhancer can completely make this collapse, or adding one can make of one form, and so on and so on. And that's what I note over here. All of these are, of course, potential signatures of cooperative processes. So the molecules involved in transcription, such as transcription factors, key co-activators, RNA polymerase II, they all have a structured protein domain, but they also have these intrinsically disordered regions. And of course, these structured domains can interact and carry out function. But the IDRs, as they are called, intrinsically disordered regions, they too can interact with each other, and through weak interactions that are not like strong chemical bonds. Now, we wondered whether in these superenhancers, if a high density of these molecules is brought together by the different enhancer elements, Could these intrinsically disordered regions interact with each other through multivalent weak cooperative interactions and lead to the formation of some of these droplet-like phases? And so to interrogate that or explore this, we construct a very simple model, extraordinarily simple, really, that these IDRs have various sites that can be modified by post-transcriptional modifications, and then these modified sites could interact with each other weakly and come apart, and so on, and multiple bonds could be formed in principle between various players. We interrogated this. By the way, the screen is getting cut at the bottom. The last part is not there because we are on Zoom. This had another line on it, which is not there. But anyway, it's OK. So I'll read them out. So what we did was we interrogated this model using basic, very simple Monte Carlo calculations. And again, this is all background, so I'm not going to show you any results, but it's well described in this old paper. What it does happen is that it forms a little phase-separated little condensate when the valency of interaction exceeds the threshold. So basically what's happening, of course, is trivial. What's going on is that if the multivalent interactions occur, the condensate forms when energy is gained by forming bonds, but it overcomes, and then it overcomes the entropy lost by confining all these molecules in one place. So of course, such a transition is first order, and so it's sharp and can explain many of these cooperative processes. As I had written here, I'm just going to give a hard time to Sarah about this. I didn't want to be on Zoom, so I'm going to say this because if we were not on Zoom, we would be able to read the slide at the bottom. Now, but this was just a, statement based on simple model. But a year later, in two different papers in Science, one from Rick's lab and one from Ibrahim Sisay's lab, you'll hear from both of them later in this meeting. And I'm going to show you data for the moment, only from Rick's lab. And what you see is that this is fluorescence data for a particular coactivator, BRD4. Here's for another coactivator, MED1. The blue is the DNA, and you see these these droplets form at these locations. And then to see whether or not it's implicated in transcription, and Ibrahim's paper does this quite vividly, but even here in this paper by Rick, what they did was they coupled this immunofluorescence with RNA fish, and so you could see that the nascent RNA transcription co-localized with these puncta that you observed. So this and more data that I'm not showing you showed that these bodies did form and were likely implicated in transcription. So but there was a problem that you would worry about if you tried to make these same molecules form these droplets in vitro. If you made these molecules form droplets in vitro, you would find that they do form, but at concentrations that are 1 to 2 orders of magnitude higher than would happen, than those that prevail in the nucleus. So the question that immediately came to people's mind is, how do they form at these low concentrations, and that too at specific genomic loci? So this was explored by a variety of methods. And again, this is part of background, so I won't say very much about it. It's all published work from some time ago, but I'll give you some description because this is a summer school. And this was work that was done by Krishna Srinivas, who's a Simons postdoc fellow at Harvard now, and Ben Sabari, who was then at Rick's lab, who is now a faculty member at UT Southwestern. So what Krishna did was he carried out some, again, very straightforward Langevin molecular dynamics calculations. But what he did was he took a mixture of transcription factors and coactivators. These transcription factors had structured domains and an IDR. Coactivators had IDRs. And the transcription factors could potentially bind to cognate binding sites on the DNA. So he first did these calculations in the absence of any DNA at concentrations that represent physiological concentrations in the nucleus. And what is shown here in this just one piece of data from this work is what is scaled size. That is, you watch what is the size scaled by, if you had DNA, how many binding sites are there in DNA. So if there are binding sites in DNA, the transcription factors would just bind there, and the number of molecules you would see clustered around those binding sites are just stoichiometric, as many as you have. So it's scaled by that, and you can see when there's no DNA, nothing happens. They don't have enough energy of binding to give up the entropy. But when you throw in this DNA, you see the scale size, which is 1 if it was just stoichiometric, jumps. And then you can do many other things that they did to characterize that a liquid-like droplet indeed was forming there. And then to test some of these ideas, Ben developed this reconstituted in vitro experiment that I do want to describe a little bit because we'll use it again a little later. And that is the following. In this particular case, they took DNA with 20 binding sites for the OCT4, the transcription factor, and they mixed that with MED1 IDR, and MED1 IDR concentrations were titrated. And then you could put these on a cover slip, and then just with microscopy, look to see if droplets formed or not. And so when you do that sort of experiment, this is what you see. In the absence of DNA, in all these ranges of concentrations of MED1, you see nothing. And in the presence of DNA, above a certain concentration, you see the appearance of these droplets. You can quantify these results by taking the imaging experiments and computing a condensed fraction, which is basically the enrichment inside the droplet compared to the light phase. Okay? So basically the way you do it is you pixelate the image, and then after you pixelate the image, you find the intensity, the distribution of intensities of light through the whole image, and you will get a mean. And then you say that all those spots where the intensity is 3 sigma or 4 sigma, you can try different numbers, above this mean, that's potentially a droplet. And then if you have 9 pixels that are connected, or 16 pixels, or 24 pixels, you can try different numbers that are connected, then you say that's a droplet. Okay? And then you just find the intensity in the droplets divided by the intensity in the rest. So that's what this thing is. It's just giving you enrichment in the droplet phase as a function of these concentrations. And you get curves like this, and then you can extract saturation concentrations, and you can see that in the presence of DNA, that's 2 orders of magnitude or so lower. Why is that? Because you get binding affinity from the DNA. It also clusters all these transcription factor molecules, IDRs, together, so they can start interacting with each other, which also allows the IDRs, the co-activators, to interact. So a question that might come in your mind is, in this case, is the DNA just a nucleator, a kinetic nucleator of the phase, or is it an integral part of a multi-component phase-separated body? And that, within the simulation anyway, you can look at, because you can compute, of course, the energy of all the interacting species in the droplet, because you can just compute all the energies of forming these interactions. And then the simplest thing you can do is just compute the entropy loss as that for confinement, which is a underestimate of the entropy because you're not accounting for what is happening to the chain. But even if you do just that, you find that the entropy lost cannot be compensated by either the energy gained from the transcription factor DNA interactions or the IDR-IDR interactions. You need both of them. And this suggests that the DNA is not, I mean, they did other things. It suggests that the DNA is not just a nucleator or a kinetic aider of the formation of this thing, but is an integral component of this phase-separated assembly. There are other things that were found, and these will become, this is just background for now. And one of the things that is very important is that the specific interactions between a transcription factor and its cognate binding site is necessary to localize these things near a particular piece of the genome. So that's where the specificity to particular gene locus comes from. And here, all the things that people have described before of DNA bending, when transcription factor binds, which recruits other transcription, all of those things are at play. But then when the valency and density of these transcription factor binding sites exceeds some sharply defined thresholds, then by this more universal cooperative mechanism, this transcriptional condensate gets assembled, which recruits many transcriptional molecules. So this is sort of the picture which ends the background section of what I'm going to tell you, and start to talk in a little bit more detail about certain issues. So the picture is that molecular interactions, structured domain binding to the DNA, IDR interactions, concentrations, the molecules have to be there, concentrated in particular regions, DNA sequence features, they all lead to a free energy landscape that can promote the formation of these phase-separated droplets. However, in papers that Ibrahim does, where he does time-correlated palm spectroscopy, microscopy, he observes that these droplets, these transcriptional droplets, are of finite size and can often be of finite lifetime. Now, if these were equilibrium droplets, they would keep growing until they ran out of material, perhaps. And they certainly would have not have finite lifetimes, unless there were other processes that were occurring. And so all of this suggests that these, and it's obvious actually, I don't think any of these bodies are at equilibrium, but this body certainly is not at equilibrium, is what we felt. So we started to explore the non-equilibrium regulation of these transcriptional condensates. There are many processes that occur during transcription that are distinctly driven far from equilibrium. But one of the ones, and this is the one I'll focus on today, is transcription itself, the making of messenger RNA and degradation of messenger RNA, and so on, enhancer RNA, and so on. So this is work that has been done by several people, the aforementioned Krishna Srinivas, and then experimental work done in collaboration by John Henninger and Oskar Oskus. Oskar is here today, so that's very fortunate for me, because when you start asking me hard questions about some of these experiments, I will just defer to him. And then in the spirit of summer school, most of what I'll talk about is published work, because this is a summer school, but in the end, I'll also describe some work that's unpublished, which is largely done by Pradeep Natarajan. So I'm going to start by saying, what is the product of transcription? The product of transcription is RNA. And so the product of transcription is RNA. RNA is the negatively charged molecules. And the IDRs of these transcriptional proteins that I was telling you about are slightly net positive. And so one could imagine that what may be going on, if you only had an equilibrium picture, which, as I'll try to convince you, is not the case. If you only had an equilibrium picture, then you could think that these are two polyelectrolytes. And one is positively charged, one is negatively charged. And so there are people in this room who are much more experienced than me in all of that, but there is a very rich history of the study of polyelectrolyte mixtures that goes back over 60 years. And in recent years, this has also been done in the context of RNA itself. And here's what happens. There's a phenomena called complex coacervation that happens, which is the following. You have polyelectrolytes that are positively charged. You start adding, titrating in negatively charged polyelectrolyte. They will form complexes with each other and form this so-called complex coacervate, because they will try to balance the positive and negative charges. You see what I mean? So just the balancing of these two charges will give you a phenomena of forming these phase-separated droplets that are called complex coacervates. Very well studied in polymer physics. What has also been studied is that now if you titrate in more and more, something very interesting happens. You titrate in more and more, then you go past the charge balance. And so now you have too much negatively charged molecules, and they start repelling each other. And as a consequence, this phase breaks apart. And so you have what in very elementary physics is called a reentrant phase transition, that you form these the condensed phase, and then everything was. And then these are pictures for RNA polymer mixtures that I think come from this paper here. One of these papers from, I think it's from Banerjee et al., where you see this re-entrant phase happen as you titrate in more and more RNA. So, does this happen? The first question is, does this happen when you take RNA, that's real RNA that is produced by an enhancer, and mediator and other transcriptional molecules? One would imagine it does, but it's a good thing to test this. And so using that droplet assay, John and Osgur, they measured these things. And these are the concentrations. The MED1 IDR that I've told you about is there in 1,000 nanometers or so. You can ask, why didn't they put the whole MED1? And the thing is, for doing all these sorts of experiments, so you know, when a theorist learns a little bit about experimental things, they like to talk about it more than theory, so that's what I'm going to do now. The problem is that this MED1, as a whole protein, is very difficult to purify in large quantities so you can do these things. And so Rick's lab over the years has shown in experiments that MED1 IDR itself, which is easier to purify, is a very good proxy for doing many of these experiments. Okay, but they also did this with MED1, but for a sanity check that it actually does this. But these data are with MED1 IDR. And you see this enhancer RNA, which comes from real TRIM28 enhancer RNA. And what you're doing here is you're titrating in more and more of the titrating in more and more of the RNA in this fixed MED1 IDR concentration. And what you're looking at here are images for MED1. Then you're looking at the RNA, and they are co-localized, as shown here in the merge. And then again, you can do the same thing, compute this partition ratio from the imaging data, which I showed you before. So you get the enrichment inside the droplet. And that is what is graphed here as a function of the concentration of TRIM28 enhancer. And you can see it here visually, too, that at first you get this, and then it disappears as you titrate in more MED1. And you're seeing this also in this quantification of the same data. What you're seeing drawn in very faint blue line here is not a fit of this data. It is simply calculating what are the charges in the MED1 at that concentration and RNA at that concentration, and graphing what is the ratio of the positive to negative charges. Okay? So the negative charges are less here and more here. And you can see that this graph passes through this with this. So this is not a fit of this data. And if you look here, you will see that roughly it peaks right at charge balance, which is 1. However, I want to point out to you that if we were to fit the data, we could fit it much better. We're not fitting this data. Okay? And if you compute the Pearson correlation between the means here and the value of the charge balance, it is not 1. In this particular case, it's 0.84. Sometimes it's 0.75, which means this is not the only process that is going on. And Amy Gladfelter is over here, who will tell you that, of course not. It's not the entire process, because she knows and has characterized that the RNA can have interactions with these charged proteins through very structured parts of the RNA structure. And we are trying to work with Amy to start to characterize those to get this additional information into this picture. But for now, this was repeated with other real RNAs. I'm not going to show you that data. It basically looks like this. And we also, these guys did these experiments with antisense RNA. So the sequence is different, but the charge is same, and you get the same phenomena. And so this suggests that although not the whole story, electrostatics is the dominant force underlying this interactions between RNA and the IDRs of these transcriptional molecules. So that's the equilibrium picture. So then one can start to think about what happens to this in a non-equilibrium setting. Okay? And so we constructed a little model for this, and I want to tell you the purpose of this model. This model's purpose was not to recapitulate quantitatively experimental data. That is not the purpose of this model. The purpose of the model was to explore the mechanisms that may arise and from the model, understand how perturbation of certain parameters that were also perturbable in experiments, what were the effects of this, and then test that prediction with experiments that perturbed exactly the same parameters. So that was the purpose of this model. Okay? So then the model was very coarse-grained in the spirit of long-standing history of physics, of using ideas that Mr. Landau has described, where the free energy functional that describes purely the equilibrium characteristics of this were described with a double well potential that involved only the protein, that is the transcriptional molecules, because that simply said that by themselves they would form this phase-separated object for high enough concentrations. And then this term, which has phi r in it, which is the RNA volume fraction, that is repulsive between the RNAs. And then this is, if you do form a condensate, then this is the interfacial tension for that. And then this is the interaction between the RNA and the transcriptional molecules. And again, in the spirit of the simplest possible model, this was expanded in a power series where there is an attractive interaction between the RNA and this, but there are higher order terms. And there are usually, in this sort of Landau theory, you can exclude certain kinds of powers by symmetry arguments. There are no symmetry arguments that we could see here. So what we did was something that might make some uncomfortable. What we did was we looked at the Jacobian of the matrix of the free energy, which says what happens if you perturb it. And then you can see the parameters that give you or recapitulate qualitatively a reentrant phase behavior. That is, as you increase RNA concentrations. By the way, certain values of these coefficients of these terms would not do that for you. It will give you different kinds of behavior. Okay? Anyway, so if anybody's feeling very uncomfortable, a little later I will show you work where all of this is done with the Flory-Huggins theory, which might make some people more comfortable. In any case, that's the equilibrium description. But these are not bodies at equilibrium. So what do we think about? So now, once the condensate begins to form, these transcriptional molecules are present there, which includes RNA pol 2. And so RNA production can start. So this is, again, very simple model. RNA production happens with a certain rate that depends upon the protein concentration here. And it gets degraded on a time scale that is fairly quick compared to the time scale over which these transcriptional molecules live. The transcriptional molecules live for hours, okay, the proteins themselves. So therefore, if the transcription molecules live forever, that means on the time scale of the observation of these experiments, by live forever, I mean they live for hours, and so for in the time scale of the experiment, it's a conserved species. So again, using very standard ideas from physical science, you can write the dynamics of the transcriptional molecules, which I have labeled protein, using model B dynamics. So this is a conserved species going down a free energy hill. Okay? However, the RNA dynamics are given by a reaction diffusion equation, which is decidedly not going down a free energy hill, going down a free energy valley. Okay? I'm used to fitness landscapes of viruses, so I call them hills. But so anyway, so they go down. So the dynamics total, because the free energy has this phi r order parameter, is not just going down a free energy valley. It is driven for, it has non-equilibrium dynamics in it, and I'll demonstrate manifestations of that. So here, what you're seeing in this movie, and I'll play it again, is that you are looking at a two-dimensional projection, and by the way, so for numerical details, these things are just differential equations, really, at the end of the day, so you can solve them numerically on a grid, okay? And you can do these in 3 dimensionals, 2 dimensions, so on. Qualitative results are not very different. Now, what you're going to see here is a projection in 2 dimensions of what is happening to the transcriptional molecules when you get this going. And here you will see the RNA, which is too dim for you to probably see. Well, that is what it is. So let's look at it again. Okay, so now look. The first one is the one where the production rate of RNA is not very high, a lowly expressed gene. Maybe this is an enhancer RNA. Okay? So then you see what happens. That thing grows, stabilizes, and has a finite size. Okay? And it has a finite size because the RNA is being produced and degraded. So there's very soon at steady state, you will have some finite concentration of RNA there. And that has not exceeded the reentrant phase transition. And therefore, it actually makes the droplet a little bit bigger than it would have been. Well, otherwise, if you had enough material, this droplet would keep growing. But for when RNA is there, it will not keep growing because of this reason. OK, now let's look here what happens when you increase the production rate of RNA. So a burst of messenger RNA, for example. Now, that's the only thing that has changed. And so you will see the RNA more clearly. But now look at what this happens. This starts to grow. And after a time, you will see that this will shrink. And the reason that happens is because at first, the burst of RNA attracts more transcriptional molecules because the RNA wants to be compensated by the positive charge of these things. But once you make too much, it goes past the reentrance and then has to settle back down to some new steady state. In fact, under some conditions, it can completely dissolve. So here, what we are graphing is from these simulations, the condensate radius normalized to whatever is the value here, to 1. And then you see as RNA production rate goes up, the size will grow, and then it will drop, and it will dissolve. So you can, so now then, is this, can we look at some of these features with experiments and see whether this is happening? First, let's look at some older experiments that were done by Ibrahim Cisse's group, which I have told you. And he measures the lifetime of these condensates. And this is the lifetime distribution that he sees of many transient clusters. And then near superenhancers, he finds these stable clusters. So these superenhancers are characterized by a high protein density, transcriptional molecule density, because the different enhancer elements all bring this together. So then, this is the calculation. These units are just simulation time units. And you can mimic the formation of superenhancers versus this by changing the protein concentration that's available to this simulation volume, because our simulation is really simulating a local microenvironment, not the whole nucleus, okay, just near this genomic loci. And you see that as protein concentration increases, the lifetime starts to increase, and then at some point it becomes stable and doesn't change anymore, as shown here. Okay, so then we said, okay, so at least it's somewhat consistent with these data. So then the next step is, can we now manipulate parameters in our model and then test by manipulating similar parameters in experiments? So one thing one could do is, what is the consequence of reduced RNA synthesis? So this is a prediction from the model, the condensate lifetime, the same graph as you were seeing here, but now you are seeing the black curve. Now there's a red curve where you reduce the rate of RNA synthesis. So it's like inhibiting transcription. So the prediction is that in these regions where the things are transient and dissolve, that is, it's going past the reentrant transition when you make more of these RNA, then there, if you inhibit transcription, the size of these droplets should go up because you have brought it closer to the low Kp than the high Kp limit. Okay? So this can be done using TC-POM, which you will hear a lot about today, tomorrow, from in 2 talks that Ibrahim Cisse will give. So for the moment, let me just say that what you can do is you can measure the cumulative distribution, and this is the time of these condensates, which is again cut off. And this black line is what you see if you do an experiment with embryonic stem cells. And then if you add DRB, which is a transcriptional inhibitor, because it inhibits CDK9-mediated phosphorylation, which allows RNA pol2 pause release, which then allows elongation to happen. So then you see what happens is that that's the red curve, and that is shifted to higher times. And if you wash off this DRB, you can basically get back to wild-type kind of behavior. So inhibiting transcription certainly makes the cluster lifetime become higher. Now, if what I've been saying is correct, then of course it should also increase the cluster size, because it goes to the low Kp limit rather than the high Kp limit, as I showed you some while ago. Same argument. And so this can be measured, and here are microscopy images. This is without any inhibition of transcription, and this is by adding actinomycin D, which causes DNA intercalation and thereby inhibits transcription. And this is DRB again, which I showed you. And if you quantify this data, this thing is about 2 to 3 times higher condensate sizes that you get. I haven't shown you the quantification. So now I want to show you that these effects can really be non-equilibrium in character. So here you see a case where you're forming this condensate, RNA is also forming, and you see this form this stable size cluster. Now, I will change only one parameter. I will not change the rate of formation of RNA. I will not change the rate of degradation of RNA. Nothing. I'll only change one parameter. And that is, so in equilibrium, exactly the same amount of RNA is being made in this system, at steady state rather. All I'm going to change is the rate of transport of RNA away from the gene body. Nothing else. And what happens? That thing will dissolve. And why is that happening? That is happening because this charge balance, so the RNA, there are 2 timescales in this problem. One is the time for the RNA to diffuse away from the gene body. And if that is very fast, this object would be at equilibrium. But the other time is the reaction time to make the RNA. So if you take a ratio of those 2 numbers, when the diffusion time becomes much longer than this reaction time, the RNA will concentrate near the gene body and not go away. So overall, the RNA concentration has not gone past this reentrant phase transition. But locally it does, which then breaks apart this concept by instability. And you can do stability analysis and so forth, which we have done, and I won't describe right now. Now, if you go and look, and then, and we estimated these, and it's all described in the supplement of this paper in some detail, then you will find that cellular parameters are likely in this range and not in this range. So this is really potentially non-equilibrium regulation of this that goes a little bit beyond just looking at reentrant phase transitions. So now, if you believe that only charge matters, which I've already told you is not an exact thing, and so forth, then you could argue that the length of the RNA should matter, because more length, more charge. So here are some calculations as a function of RNA synthesis rate, normalized condensate radius, normalized to this point, which was just 1 point in our calculations. And you see for short RNA, as you make RNA synthesis rate go up, you will increase the size of this radius. For reasons I've described, the RNA adds more charge compensation, and you form these condensates better. For medium RNA, it should go up and then go down. Again, I showed you that recently also. And for long RNA, this should very quickly cause degradation because you'll quickly go past the reentrant point. So this can be tested in cellular constructs, because what you can do is you can make cell lines where, well, you as in Oscar and John can make cell lines where they had a DOX-inducible promoter that makes what they call feedback RNAs that is right adjacent to another gene that can be reported out using luciferase. I should point out that they did many controls. That is, they put this and this gene as convergent, divergent, but most importantly, they also did cis and trans. And they found that as long as it was cis, the phenomena that I will show you happens. Okay? And so now here, so then you can read out this luciferase, and then since this is DOX-inducible, you can turn on the rate of this RNA synthesis mimicking this axis. Okay? So this is what we wanted to perturb, and you can put in different RNAs of different length. So then you can perturb this variable that we perturbed in the calculation, you can perturb this variable. So here's what you see. This is what you're reading out with luciferase. This is the DOX dose, which you cannot read. And then you see what happens. This is the shortest RNA, 185, longer, which mimics this one, and then the longest with this very unlucky number 1313. I don't know why they picked it. It goes this way. Okay? So all of this is suggesting a picture, and suggest is the word, not proving, suggesting a picture where short, transient enhancer RNA that are made in the vicinity of the coding genes promoter, they can promote the formation of these condensates, and then a burst of messenger RNA, which is both longer and more long-lived, could potentially then dissolve this condensate, saying that this could be A, not D, a contributing mechanism to the many others that play a role in deciding bursts of transcription. Okay? Now, this experiment, where you have the RNA being produced vicinally to the promoter gene that you want to actually report on, producing these data, made us start to think about another puzzle. And this is all probably not appropriate for a summer school presentation, but I'll go ahead and do this. And the things we were thinking about, there's very many puzzling observations about long non-coding RNAs and their functions. And there are many speculations, data on what these functions might be. I'm going to add a new speculation based on theoretical calculations, not yet data, on what these long non-coding RNAs might be doing. And the work stems, as I told you, from this experiment with one motivator, and the other are many puzzles that have been noted in the literature. These long non-coding RNAs are often present in genomic and spatial proximity to protein coding genes. And very interestingly, across animals, the distance, both genomic distance and spatial distance between these two loci seem to be very conserved. It's an interesting observation. It has also been noted that these mature long non-coding RNAs can localize near the RNA, long non-coding RNA locus after they are made. They have a measurable effect on gene expression by the nearby protein coding gene. And in this paper from Eric Lander's group, they showed that it can both enhance and repress the gene transcription from the protein coding gene. They also found that if you go and change the sequence of this RNA in the long non-coding RNA genome locus, then as long as it's transcribing, it has the same effect. So suggesting that the transcription of this RNA and not its actual identity may be important. And this is consistent with the idea that while these distances are conserved across animals, these sequences are poorly conserved. So we began to wonder what may be the origin of all of these things. So again, we'll make a model in the spirit of Occam's razor, very simple model. And the model is basically this, that there is a protein coding gene locus. There's a low, LCL is just long non-coding gene locus. And they are present here. I told you that in that old paper, the DNA attracts the transcription factors. Very coarse-grained model of this is that there is a well, free energy well, that brings the proteins, the transcriptional proteins for the protein coding gene over there. And I told you that the long coding RNA is localized near the gene locus, its own gene locus, so that's another well. and they are a certain distance apart, LP, which we shall change. You can then make, first of all, a free energy of this. Now, instead of Landau, we can use Flory-Huggins, just to show you that very similar things happen. This is the well for this one. This is the well for that one. That's the surface tension term. And now let's first look when there's no transcription of the long non-coding RNA. All that's going on is that they're there. Okay? And then this process, transcription molecules start arriving at the protein coding gene, is what is turned on. So then in this time scale, then, the way we are looking at it, then the RNA and the transcriptional proteins are conserved. They're both given by model B dynamics, which you cannot see. And then you sort of watch what happens. So here's what you see. So this is how much protein you have put in the volume fraction in your simulation box. And this is the enrichment at this site compared to this average. And this is when there is no RNA localized at the RNA locus. So this doesn't do anything. And this is when there is RNA localized. And you can see that you get transcriptional condensates that are bigger. And now you know the reason why. It's just the same as I described to you before. But it's a little interesting that, because this phenomena, this enrichment, let's say I take this point, and I change now the distance, I change the distance between these two. And you see this is between the two loci, and then it'll go away. And the reason is that these two wells, or these two distributions, have to overlap for the RNA to feel the presence of this, and therefore, through these dynamics, go over there and compensate for these charges. If it's farther away, it won't happen. So this is just very straightforward thinking about this. But one wonders then, could this be a reason why these distances are so conserved, even though their sequences are not? Finally, what I want to talk about is simply what happens if I now have both of them transcribing, which is the case that Lander's experiment looked at, where the long non-coding RNA was being transcribed, and this was being transcribed, and they looked at whether or not it affects protein coding gene expression. Sometimes it suppresses, sometimes it enhances, and sometimes the sequence of this RNA is not as important as there's transcription going on. Those were their findings. So the model is very similar. Now you have two RNA species, the long non-coding RNA and the messenger RNA that can be produced from the protein coding gene. I've told you about these terms before. This is just Flory-Huggins for a three-component system. Very straightforward. You give them as homework problems in grad school. Okay, so now the dynamics, however, are different. On the timescale of these processes as before, the transcription proteins are conserved and given by model B dynamics, but both of these RNA species are transcribing and degrading, so they are reaction diffusion equations. And now the dynamics, unlike what I was showing you here, now the dynamics is certainly not just going down a free energy value. So what do you see? Our time is running short, so this will probably be one of the last things I'll show you. And what this is the fold change in amount of RNA made, the messenger RNA that is being made. This is the long non-coding RNA transcription rate. How quickly are you making long non-coding RNA? And the different graphs are for different rates of messenger RNA transcription. So if this graph is small, if this value is small, it means that this gene is expressed at low levels. And if this value is large, it means these genes are expressed at high values, highly expressed genes. And what do you see? When the gene is expressed at low levels, the messenger RNA, then if you increase the long non-coding RNA transcription rate, you see an increase in transcription, and then ultimately it goes down. And now you know why, because of everything I've told you. It's simply that this RNA is now adding to the negatively charged molecules that are recruiting more and more transcriptional factors. You're getting more and more transcription. And ultimately, of course, if it becomes too fast, it goes down. But notice that if the gene is highly expressed, then this long non-coding RNA is only going to disrupt the transcription condensate, and you will see transcription go down. So the question is this an explanation potentially for why Lander's experiment was seeing both increasing of expression and decreasing of expression depending upon the gene? I don't know. So the question is, now we are hoping that either our collaborators or others will, and hopefully our collaborators will do experiments to test these ideas about whether or not long non-coding RNAs, in addition to many other functions they may have also this function, and whether it is truly an explanation for the results not only of experiments, but also this evolutionary data on the proximity being conserved across metazoa. I want to close by saying I can't really give a talk without talking anything about the immune system, so I must bring it a little bit in as obliquely as possible, because this is really not immunology, this is cell biology. But here's an interesting piece of data. All of this is from Jay Groves at Berkeley. And this is all cut off, but I will explain. What he's seeing again here are the T cell, is actually an interface between a T cell and a supported lipid bilayer. So it's a fake antigen presenting cell, okay? But it has peptide MHC. Peptide MHC stimulates the T cell receptor, okay? That's the molecule experiment. And they bind to the T cell receptor. So what you're seeing here are these particular peptide MHC, don't worry what it is. And every time you see this bright spot here, that is a TCR peptide MHC that has bound. And so Jay's lab can then, through single molecule spectroscopy, follow what this TCR peptide MHC is doing and how long it stays alive. Got the idea? Because it comes up harder, ultimately. And so he can measure the lifetime of this TCR peptide MHC bond, individual TCR peptide MHC bond at this interface. And then, without telling you all the details, as signaling goes on, after that first binding event, one of the key molecules is the molecule called LAT, which assembles a big signaling complex, okay? And it is known, starting with Ron Vale's data, that this LAT can form these sorts of condensates. This is not a liquid-like phase. This is a percolated cluster. That I know by having done we've done some work on this. But anyway, this LAT forms, and what he can watch is the time, from the time that this binding event occurred, how much time does it take to form this cluster? And that's the data here. So you see this cluster, it takes some time before it forms, and then it dissolves, just like these transcriptional condensates dissolve if the rate of transcription is high enough. These are also bodies not at equilibrium, clearly. But there's even interesting, more interesting puzzle. You cannot see this here, but this is the delay time distribution, the distribution of these delay times. And they're widely distributed, and this distribution does not depend on the lifetime of that TCR peptide MHC bond. What does depend on the lifetime of that TCR MHC bond is the probability that you will get these to form at all, but not the wait time distribution. So these are interesting puzzles, clearly non-equilibrium puzzles, coupled with a phase transition that is not, I think, might take not liquid-liquid phase transition. And so we started to play a little bit with this as well, and maybe some of the students can play with this and solve this problem before so then we won't have to think about it much. So you can think of that as a nice tutorial, recitation problem, if you like, for this class. Anyway, that's it. I'm done. I took exactly 60 minutes. So thank you.
So let's raise your hand. Let's have a question. Please, over there. Your hand was up first. Stand up, please.
Hi, I had a question. Like in the simulations, you showed the reaction.
Identify yourself and where you're from.
My name is Atreo. I am from Texas. I'm in Dave's group. my question was, in the reaction diffusion simulations, two of the non-equilibrium effects I think you showed was the condensate size burst, like increasing in a burst and then going down over time, and like forming a stable condensate, and then with the reduction of the diffusion of the RNA, the condensate like shrinking and dissolving. Is that observed? Like, are there any experimental observations for those two effects?
Well, first of all, let me tell you, both of those phenomena can be gotten without being driven strongly out of equilibrium. Okay? The thing that I was mentioning, so let me just clarify this a little bit more because I didn't put an actual figure about this. But for example, growing and dissolving, that can happen simply because you make more and more RNA. At first, you're promoting this effect, and you can recruit more transcription molecules. And when the overall concentration throughout the whole droplet becomes more than a certain amount, then it will dissolve. So those can be obtained just by steady state behavior, okay, sort of steady state behavior. Now, have they been observed? What has been observed is what I showed you in the experiments, where that condensate formation, if you view that as a proxy for transcription, because that has been seen in other experiments, like the ones I briefly alluded to, then by changing the RNA that is coming in there, you can change the transcriptional output.
Okay. So
that is what has been done. For the non-equilibrium part, and I do want to clarify this because I obviously made a mess here. So that what I was trying to say is that suppose you keep the production rate of the RNA and the degradation rate the same. Actually, okay, so you keep this the same. And so from a steady state behavior, these things should behave the same as you change the transport coefficient, the rate at which the RNA moves out.
Yeah.
But on the other hand, if the transport rate of the RNA is small enough, d less than some threshold, and at the threshold I will tell you, then the RNA will mostly localize near the gene body, which actually happened. I was just talking to Mitch Gutman, who knows more about transcription, who has probably forgotten more about transcription than I know. There he is. And he thinks that that's likely what happens during these things, potentially. Then what you will see is that even though overall nothing is changing, but locally it will break charge balance, and you will see this thing happen. But in fact, if you do this and contrive just a droplet without the DNA and all this kind of stuff, you can even see very interesting thing happening in the middle of the content. You get a vacuole to form. But those I don't know have any biological relevance. It's interesting instability analyses, but that I don't know. And the threshold is very simple. You have two time scales. One is the reaction time at which you're producing this. So if it's just production of this, it'd be 1 over Kp times the protein concentration. So that's that. And the Td is just the size of the condensate divided by d. And when this number, Td over Tr, becomes less than 1, you will see these non-equivalent effects. And in that paper, I don't want to recapitulate right now, we estimated these numbers for transcription, and it seems that the actual cellular parameters are in this range.
Okay.
Okay. There were several arms up. There's a question over here.
Over here? Okay.
Hi, I'm Shady. I'm a postdoc in Ioana Waisoka and Daniel Jarosz lab at Stanford. And I'm wondering about what about the 20 to 30% of transcription factor that have negative charge? And I have a second question about are we limiting our knowledge to the IDRs that we can purify and they are charged in nature? Because most of the IDRs we cannot purify and they are not charged at all.
This may be, but the ones we have looked at, both questions are very legitimate questions. But I would answer in two ways. That first, what we have looked at, which is the transcriptional protein IDRs, such as MED1 and BRD4, they are positively charged. not because of purification, but they are. In fact, you can lose some of this behavior, not this particular behavior, but just forming the condensate and so on, if you take the histidines in the MED1 IDR and convert them to alanine, for example, and not histidine, the serines, and also the histidines in another context. But so you can manipulate those charges and see effects of it. So that one thing is that in the context of this specific system is what we have looked at, but more general things definitely happen. That is why I kept saying throughout this, it is a contributing mechanism, not the mechanism. And the other part I really want to emphasize is that RNA has structured interactions, and we need to think about those carefully in this context, which we are trying to initiate with Amy Gladfelder and so on. So that's the answer. It is not a negation of anything that you say. There were other questions here. Margaret has.
Yeah, maybe
please
just a
stand
quick.
up, please,
Oh, okay.
and identify yourself.
So Margaret Johnson, I'm at Johns Hopkins. And so I wanted to follow up just about the diffusions because like you showed, if you diffuse slowly or your mobility is very slow, then you can have these things dissipate. But you also showed that as you made the RNA get bigger and bigger, you had this trend due to the electrostatics. So do you think that that's just going to dominate over the diffusion? Because of course you get bigger and bigger, your diffusion also will slow.
Yeah, so they'll go in the same direction though, so it'll be difficult to see if you make it longer or you make the diffusion slower, it'll go in the same direction.
Oh, okay.
Right, because diffusion slower means you'll concentrate in local regions, which will have too much negative charge there then.
Oh, right.
And if you have longer, you'll have the same effect. But the other question that you might want to ask for is that if you're seeing this, then can you make the effect go away by making it shorter? Because that would but add more molecules, make the transcription rate higher, but make them shorter, and they would be diffusing more faster, and see if you can balance these effects. We haven't done that, but it's possible.
There's one here or over here?
Hi, my name is Alex. I'm a PhD student at Stanford in Dan Yarosh's lab. So first question was actually a follow-up to Shady's question regarding the charge. So for the ones that you've looked at where they do have a positive charge, we know that the sequence can diverge greatly, like through the course of evolution of an IDR, but I'm wondering if that charge is conserved.
So what I can tell you is an estimate, not based for this, but for the long non-coding RNAs. And again, there are people here who may know a bit more than I, but when you look at the sequences not being conserved of these long non-coding RNAs, if you do very rough analysis, which we did some years ago, maybe Pradeep remembers better than me, the charges are not exactly conserved. But
oh, I'm sorry. I meant the sequence of the IDR of the TFs.
No, they certainly will not be conserved across all functions.
Right, so the sequence isn't conserved, but sometimes the IDR domain is, from way down all the way up. But is the charge conserved? So even though the sequence might diverge, does that?
I don't know the answer. Okay, I don't know the answer, but my guess would be if they are doing similar functions, like in transcription, they're probably still slightly net positive, but I don't know. The place to look is in Judy Foreman-Kay's, is that right? Yeah, Judy Foreman-Kay's paper, because she has done a lot of analysis of these charges. And there is some unpublished data, so I should not talk about it much, not from our lab, otherwise I would talk about it, not my collaborators' labs, but Ben Sabari's lab, where they have been looking at this. And it appears, let me just leave it vague because he might get upset but it appears that at least some features are conserved in the sense of blocks of length this and how alternating they are, and those things seem to be somewhat conserved.
Thank you. I had a separate question about, and I might have missed this. So in the case of the super enhancers where there's just a ton of proteins that are there, I'm wondering if, so all of those protein-protein interactions that could stabilize a phase, are those more robust to RNA production, and is that,
For sure.
yeah, okay.
For sure. And that is what you saw in that graph I presented with the amount of protein in the microenvironment. And you saw that as I added more protein in the microenvironment, you became a stable cluster. And that's true in Ibrahim's experiments when he does that. He sees that those near the superenhancer largely form the stable clusters.
So do you plan to simulate it all, like different protein-protein interactions, like heterodimer?
Yeah, so those are complexities that we do want to build, but I would rather first build this thing with the RNA into this, because I think it can play a very important role, because there are RNA binding proteins there too, which we have not done, so that would be a true multi-component mixture of these things, and we do want to look at that.
Well, thank you.
But we haven't done it yet. Manju?
Here, let's go here first. and then-
Oh, okay.
Hi, I'm Shumita from Rice. So I had a question about, so if I understood correctly, most of the forces in your computations, they are derived as sort of gradients of potentials, right?
No.
But the potential is changing, with the free energy is changing with time?
Yes, because of the reaction
Right.
diffusion equations.
Right.
Yeah.
So, but there is another paradigm where forces are derived from like nanomachines, which are pulling and pushing at the microscopic level. And so do you think those forces are at all relevant, or you think in your computation you are including them?
In the time scale of transcription at the local gene, in the time scale we have observed, I think that these reaction diffusion forces that do take you to achieve the same goal, take you out of the equilibrium, are probably more dominant. But are those forces not there? I don't know. For example, there could be chromatin bending that happens, or things like that that would add other features. We think that it happens likely on longer timescales than what we are interested in here.
I see. Okay, thanks.
Right.
I'm Don Rio. I'm from UC Berkeley, just visiting. I wanted to ask you earlier about your low complexity domain experiments. There's a paper in Molecular Cell in 2020 suggesting that low complexity domains can either contribute to or direct site-specific DNA binding by transcription factors. How does that fit into your...
We have ignored the binding of these IDRs to structured domains, okay, in this picture. Again, we think that the dominant effect is these weak multivalent interactions, but undoubtedly they can happen, such as wanting to put more different proteins in it and so forth, and they will influence some of this behavior.
Yeah.
But, and quantitatively, they will definitely impose it, but it's there. But we have not, we think that the IDR-IDR interactions are probably dominant in this context.
Right, there's no real evidence, though, in vitro that they can actually recognize specific sequences.
You mean the...
The IDRs.
Yeah, so also in vitro, these IDRs, just by these
Yeah.
weak interactions, form these complexes, as I
So there
showed.
must be something in addition that's happening in vivo. If IDRs can actually direct transcription factors to specific sites on DNA and cells.
Yeah, so
That's
I mean...
what that paper implies.
Yeah. There are
Yeah.
additional features to this
Yeah.
that we have said that specificity is largely coming from the structured domain binding to the DNA.
Right, yeah. I had one comment, which is,
Because you see, if that piece was not dominant, the structured domain binding to the DNA,
Sure.
You'd find it everywhere.
yeah, it just seems to be an additional
No, for sure.
function that may play a role in vivo.
Of course.
And I just had one comment about...
Well, so the formation on these particular DNA loci, you can disrupt by changing the density of these binding sites in vivo.
I see.
Okay?
Yeah. Just one comment about the role of RNA. I think there's a 2013 Genes in Development paper from Tom Check's lab that shows that if you add RNA to the low complexity C-terminal domain of Pol II, it can form a condensate. So there was evidence some time ago that RNA would allow condensation.
There's evidence before that in polymer physics literature. Denny's and co-workers did that, as I pointed out. So this is well-known 60-year-old physics. So.
Question, there's
a few.
go there, and then we'll go over here.
Hi, I'm a postdoc at in Michele De Pier's group at Northeastern. So, so I was wondering, in the Flory-Huggins model that you talked about, did you explicitly consider the charge interactions, like the electrostatics?
Screened electrostatics, because there is salt.
Okay. Okay. So you have the multivalent pairwise interactions, and on top of that, you have the
Screened electrostatics, yeah.
Okay. Okay. Great. Thank you.
Let's go over here.
To Manju or?
Arup, come over here where you can be in the light for the people who might be watching us.
I'll pass it to
Okay.
her. So, yeah, I'm Manju Hingorani from the National Science Foundation. Arup, I think you started alluding to this in other answers. But my question is, are there differences between small RNAs and long RNAs that might result in differences in their regulatory effects, maybe make one or the other better regulator in certain gene expression regimes? Could you elaborate on that?
Yeah, so I will largely speculate based on our results rather than firmly. So one thing that we think is that maybe the enhancer RNAs, which are shorter and more transient, nearby they get these transcriptional condensates to form, whereas the longer mRNA transcription can dissolve this. But in the break, I was talking with Mitch Gutman, who thinks that there are other things that may be happening during transcription, that you get these shorter RNAs to form first for various reasons, and then those could be the stabilizers. And then there's an interesting question then, that does the length of the RNA, its rate of production, and so on, naturally limit how much transcription you can get at a time from a gene? And those are open questions that need to be explored.
Right, stop sites and reasons for making shorter transcripts might also be regulated for this purpose.
For sure.
There's another question right back here.
Seychelle Voss, MIT.
Hi.
Yes. Thank you guys for organizing this conference. It's great. I really enjoyed learning about the long non-coding RNAs and having the distance requirement and not a sequence requirement. I was wondering if you guys had looked at all into directionality, if that matters, if the directionality of the long non-coding transcript versus the mRNA matters.
We have not, but we should talk and see how we can explore this.
Yeah.
But we could certainly do that.
Yeah.
Now, John and Oscar did, in the context of that contrived system, because they put this RNA gene, that the gene of making the RNA and the reported gene, they did do both divergent and convergent. I pointed in the wrong direction. Anyway, and they got the same thing. But if they put it in trans, then this effect goes away. But that's pretty straightforward to understand why.
Right.
Over here. And then we'll come back there.
Hi, I'm Michele Di Piero, and I'm in the physics department at Northeastern. So actually, I wanted to follow up on Seychelle's question. First of all, I gotta say, from a physicist point of view, I really thank you for your talk, because it was a good introduction and things that were less familiar. And about this long non-coding RNA, it's more than a question. I have a curiosity. I would like to ask you to elaborate a little bit on how do they establish that the distance is conserved? You mentioned that was metazoans. So, could you tell us a little bit more about that?
Yes. The papers are cited. I can also give you the papers there on there. So basically, what they look at, actually, the person who can give you a more detailed answer than I is your neighbor, probably. If you give him the mic, he will give you potentially more detailed answer than I, because this is the thing of the discussion. Yeah.
Stand up and identify yourself.
Sure, yes.
So I showed his picture earlier. So
yes. Hi, I'm Pradeep. I did some of the long non-coding RNA stuff with Arup. So to answer Michele's question, I guess, so the analysis was something called a syntonic analysis, which people do, which is like you can look at multiple different organisms, and then you can take homologous genes across organisms, and then you can look sort of what are the different long non-coding RNAs that are there, annotated according to some database in the neighborhood of those homologous genes. And then you can see how the distances between, let's say, homologous gene in a human and a mouse is to its, let's say, its nearest long non-coding RNA. And then you could do that across multiple homologous genes and multiple long non-coding RNAs, and then you could look at like what their distances look like and plot the distributions. And also, you could kind of look at, for a given long non-coding RNA, whether it lies in the same genomic neighborhood as defined by its neighboring protein coding genes. And then you can kind of see if the distances between PCG and its nearest long non-coding RNA, and also for a given long non-coding RNA across multiple organisms, its corresponding nearest PCGs, if they're conserved or not, and like what is the distance between them, and that's kind of the analysis that they do.
And obviously you can do the sequence analysis the same way and find the sequences, however, are not conserved.
Yeah.
And the other way that they look at a different question, which I also alluded to there for the sequence business, is an experiment where you can go and change the piece of DNA that makes that RNA, but it still transcribes the RNA. And as long as you can scramble the sequence, but as long as the charges and so on, but they didn't know what charges, as long as the length and so forth is conserved, then you see very similar behavior.
We'll come to you in a moment. There's a person right behind that just saves steps. Please stand up and identify yourself.
Hello, everyone. I'm Henrik, studying physics here at MIT. I was wondering, this mechanism for regulating transcription, this proposal and these observations, how does that play in with other ways of taking decisions in a cell? So this is one knob that you can dial to turn on and off genes. A, how often does it happen in the cell? And B, as a way of regulating transcription, which types of decisions would this mechanism be good or bad for?
I don't know how often it happens in a cell. I think if I were to speculate, I think it's a question worthy of the study. I don't know how often it happens in a cell, but I would think it happens more when you have highly expressed genes. That would be my guess. But I will defer to the session chair and Rick Young, who, again, know more about transcription than I do, to speculate on this question.
So I don't specifically understand the question. So can you be a little bit more specific about the first one?
Yeah, the first one is just how many genes are regulated in such a way as this?
Essentially, well, let's say you got 20,000 genes, protein coding genes, that's what we're talking about. And in a typical cell, 10,000 will be expressed. And in other cells, the other 10,000 will be expressed. There will be something on the order, let's say, Mitch would be better at this than I do, but let's say 3,000 that are expressed in all the cells. They tend to be those genes that are doing metabolism and other things. And then the other 6,000 will vary from one cell type to another to various degree. We think all of them are primarily controlled by transcription factor genes expressed in those cells. So there's a concordance between a set of transcription factors and the set of genes that are expressed. And then other cells will have different transcription factors and genes that express. Obviously, there has to be a homeostatic process by which transcription factors control transcription factors and maintain that cell state. Transcription initiation is a critical step, but every process along the way, there is, again, more subtle and, in many cases, environmentally controlled expression of genes, stress, nutrients, and other things. They tune them up and down. Growth rates are tuned up and down. So, this is, it's an interesting question, but that's the way I would view the regulation of genes.
And what fraction are turned off by the mRNA transcription as the principal mechanism, I don't think we know. We know that at least in cellular constructs, it does happen. And then the trick now is to do these experiments with the long non-coding RNA problem that I showed right at the end, because then you can regulate what the long non-coding RNA is doing by changing its sequence and so on, and see how frequently it matters to the actual transcription. An extension of the Lander paper manipulating what's happening in the long non-coding RNA. So that would be the experiment to see how frequently it happens, at least in that context.
I do need to say something that most people in transcription don't pay attention to, but I think it's pretty clear. Transcription occurs by bursting. So you could have a cell with a gene in a cell, give you a burst of 4 or 5 polymerases transversing the gene, and then for the next 10 hours, that gene will not be transcribed. So you get a burst of transcription. When that's, I think most people believe the majority of genes are expressed that way. I wouldn't say all of them, but I do think a majority are. So that's when Arup talks about non-equilibrium processes initiating and terminating transcription. It is initiating and terminating that burst, and then the number of polymerases have to reflect either the size of the burst or the frequency. And there's some argument in the literature about those two things. There's a question back there.
Hi, I'm Ben from MIT Chemistry. Great talk, Arup. Really give me a lot to think about. So I want to follow up on the question where you were saying that for the condensate between protein and RNA, they seem to have a well-defined distance that seems to be very preserved. So, and you're saying that's also kind of a sequence independent, which was a little bit kind of counterintuitive.
That's just the experimental data.
Right. So then I guess I was thinking then, if that's the case, then in the cell, you probably have a lot of those RNA condensates. So then, I mean, then the protein might actually also, wouldn't the protein condensate engage with other sort of RNA condensate, which then sort of actually leave your distance to be less well determined?
So at least for the data that I know, these long non-coding RNAs are often in the vicinity of that gene, of the gene that produced this. So what you could say is how are they there? And there could be many mechanisms. One is that it is held there by some reason, but the other could be what you are saying, that other proteins, that RNA binding proteins, could bind to this RNA and localize it there. This is one possibility. And I don't know whether it's true, but it's one mechanism where how that RNA could be held there by these RNA binding proteins. And then when other things happen in its vicinity, then it could regulate other things too. But I think the more interesting case is the one where the long non-coding RNA is being transcribed, which is the last part that I talked about. There it's not just hanging out there, it's being transcribed right next to it.
I mean, I agree with that. I was sort of thinking, but then that would have basically happen for any kind of RNA condensate, right? You know, it doesn't have to be this condensate from a particular chromatin region. It could be like any region.
It could be, yes. And so the question is, where are these RNA depots? There could be ones that are hanging out in various places, and I don't know.
Right. Okay. All right.
Just to add a nuance to that, it's clear that some transcription factors that bind to DNA also bind RNA, and I suspect you'll hear more about that later. So they can localize RNA, potentially localize RNA in a region.
And Rick will talk about that.
And Rick has already published in YY1 that data. Let's see, who was up? It doesn't really matter. Start there and we'll go down the line.
Well, very, very exciting work. And I think also ultimately quite, let's say, controversial. You have really two mechanisms going on in your model. One of them is the phase separation mechanism that's been very popular. And then in that mechanism, the size and structure of things is determined by this sort of surface tension effect.
No, it's determined also by the non-equilibrium nature.
I'm sorry, I'm talking in the pure phase separation.
Oh, pure phase separation, okay, sorry.
In your case, the other effect is something that's analogous to what goes on in combustion. You have a reaction which is autocatalytic, which is of course why the study of flames is a big part of chemical engineering, so it's probably not a surprise that you came up with this. Now, combine both of these, but it's not clear to me that, for example, the surface tension effect played any major role in determining the size, but that was a detail here, and so you sort of imagine this is something like the problem of what determines the size of a flame. So maybe it wasn't clear to me in the experiments whether there was an experiment that just sort of tuned the rate of combustion, like the way we tune temperature.
Yes.
You had an example, but it was seemed like it off.
No, it was not. It
Continuous manner.
was changing how much was being made. So let's see.
Okay. It's hard to show.
Yeah.
So, I think that there should be fairly detailed predictions from this.
Yeah,
so
those are, they are not detailed quantitative predictions, but they are detailed qualitative predictions, which is this. So this axis here is the rate at which you're making RNA.
Mm-hmm.
That's the prediction part. And this axis here is the rate at which you're, is tuning a very similar parameter by changing the amount of docks.
And what are the vertical axes? I can't read them.
The vertical axis here is the size of the condensate, which in other papers we have, Rick has shown correlates with the, and Ibrahim too, correlates with the amount of transcriptional output. And what we are measuring here is simply the transcriptional output from this luciferase gene.
I see. It's still very hard for me to see, but the only experimental result is the one in the lower right corner. Is that right?
Yes.
Okay.
So far.
Hi, Bram Prevel. I'm a postdoctoral fellow in the Earnshaw Lab, University of Edinburgh, UK. I was kind of triggered by one of the questions you had at one of your end of your slides, which is kind of development of key experiments. And that's kind of my question. So what do you think is key for us to bring together our in vitro understanding of phase separation in these processes and our in vivo understanding? Is that like what you already hinted, like, we have chromatin benders and remodelators, and I think often in vitro experiments are done with naked DNA.
Yeah. So Oscar, who left in Rick's lab, tried to do this. In fact, we report it in one of our papers as sort of. He tried to create, like, I think Phil can tell you who the first person to do it. I think Roder was the first person to do this, but he tried to, or maybe Phil, I don't know, but he tried to recapitulate transcription in vitro, because then you have many of these features happening there. And you see signatures of these phenomena, but it's difficult to say with surely what is going on, because unfortunately, when you do this in vitro, the transcriptional output is so small that it is difficult to really do much with it. But if that could be improved, that would be one way to really study that in vitro.
I would answer this because I spent 25 years doing in vitro transcription. Those systems are some reflection of what happens in vivo. And you can actually change conditions in many in vitro systems to get a phenomena that you anticipate or have seen footprints of or indication of in vivo. To me, I think the most data that will be the most valuable will be single molecule imaging of RNA and protein at different promoters in different cells at a moment. So you can see things form, disappear, stay, influence, build, and suppress. So that technology, I think, is going to be the best and strongest technology to actually probe these processes in vivo. That's just my opinion. And that's why Abraham Sissi is going to talk here. Next, and then Amy.
Hello, I'm Iraj from NYU. I also wanted to ask about this distance question that a few other people had asked earlier. So if I remember correctly, you were showing us that in your model you have this screening length that essentially sets the distance, right? So I was curious if the actual lengths that are measured...
No, it can be more than the screening length. It can also be the length over which the RNA is delocalized near that long non-coding RNA genome.
Okay.
Locus.
Well,
so it's a mix. It could be that too, not just the screening length.
Okay. Well, it looked in the figure you were showing, it kind of looked like it was set by this parameter sigma that you had in your model.
The sigma is yeah. Let me look at the figure itself.
Yeah, that would be nice.
Sigma is just the distance of these two things. And so that width could be set not just by the screening length of the interactions, but by how localized is the RNA in the vicinity of that long non-coding RNA gene.
Okay, I guess I got confused by the fact that
yes,
in your equation you also have sigma
right.
for the interaction.
Right.
But I guess, okay, then I was
So
just...
that's why the sigma appears here in just localizing it over there.
Okay. So then I guess my question is, if this distance is found to be conserved across all these species, then what's physically setting sigma, and why would we expect that to be conserved across species?
Yeah, so then the question is how localized is this RNA just around the long non-coding RNA gene, right?
Sure, yeah.
So that's the thing to look at. Sure. Is it about the same distance or within order of magnitude of the distance between the two? Yeah, that could be, we could look at that.
And do we know that to be?
I don't know.
Oh, we don't.
I haven't looked.
All right, okay, thank you.
Amy back there.
Hi, it's great to be here. My name is Amy Gladfelter. I'm from UNC Chapel Hill, and I had a question and a comment. So my question comes actually from the graph that you did have up just a second ago, which was the length dependence experiment, the DOX-inducible luciferase.
Oh, okay, just a minute.
And I guess it's interesting that really that the 1313, where you see kind of the maximum negative activity, basically is quite short, right, in the scheme of messages, right? And so I'm wondering if you can just speculate or what you guys think about that. Is that because of the context of where this is being expressed, or the limitations of the artificial promoter? And actually, could you use that to kind of identify modulators of
Yeah, so for example, if you looked at the mRNA that is being made here, and if you threw this feedback business out, okay, then you would find that the length required is longer.
Is what?
Is longer.
Uh-huh. Okay.
And the reason is simple, because they are not prepared right in the beginning. So in the beginning, you're getting small amounts of these, not the whole thing simultaneously.
I see, yeah.
So you can manipulate, so if you looked at just message, which was the calculations all that I was doing here, they are, if you just look at message being produced, then you will see that the lengths will go to longer lengths.
I see, okay.
But the experiment hasn't been done changing the message.
Right.
So that's an interesting experiment.
Yeah. Okay, great. And then just a comment on long non-coding RNAs and lack of conservation. I think it's just important to recognize that there's elements probably of conservation that we don't yet know how to identify based on structure, that are not currently identifiable because we don't know what patterns to look for in terms of conservation. And so while there may be some functional substitutions that can be done, such as in that Lander experiment, I think it's important to recognize we don't yet really understand that. Yeah.
We are at, I think, the program moment in which we're supposed to take a break. I just want to say, as being a cell biologist around for some years, it is so exciting to have theory directly play on experiments in real time as we're discovering how these processes work. We've had phenomena for the last 20 years, 30 years, bursting and other things and non-coding RNA. But now we have, and others will have other theories that will actually be able to lead further exploration of these facts. And having, colleagues who are physicists and others, as I'm preaching to you, is just an enormous addition to the field.